I welcome you all to my YouTube channel. Today we are going to study sexual reproduction in flowering plants. Okay, so I have brought you a flower where we can identify all these four organs. Okay, the greenish appearance when you are going to see here. Okay, this greenish appearance we are going to call this as sepals or calyx. Okay. This is going to protect this flower in bud condition. When the flower which is not yet matured, this calyx is going to protect it. And this colored part what we are going to see, we are going to call them as petals or corolla. Okay. And next the yellowish color. What we are seeing here, yes, I am going to separate them one by one. So this we are going to call them as androecium or stamens. Okay, when we are going to separate out all this thing, in between there is a gynoecium. Now I am going to dissect these all things separately and I am going to show you. So when I have separated it, you can see these were petals, I have separated them. Then these are the androecium. Okay, this yellowish part we are going to call it as an anther, and this we are going to call it as a filament. Okay, there are totally 1, 2, 3, 4, 5, and now these 5 anthers I am going to remove. Now I am going to get a gynoecium here in between. Yes, can you see? I have removed them, and in between I have got a gynoecium. Okay, and this gynoecium here bulge part. The bulge part we are going to call it as an ovary. Okay, this a thinner slender part, a stalk like part which is having a feathery texture. Okay, this we are going to call it as a style. Okay, at the tip portion we are going to call it as a stigma. One by one I am going to draw each and every part in the further session, and you people will be understanding it clearly. And here also I have brought you a very little tiny flower where you can see the calyx and white colored corolla, green colored calyx, white colored corolla, and when you are going to turn the flower very beautifully, you are going to see the androecium. Yes, yellowish colored androecium. And when you are going to dissect this tiny flowers androecium, you are going to get very little tiny gynoecium too. So friends, you can try all these things at home. See, we are having younger flowers in the center, matured flowers towards the outer side. Okay, these all colors have been changed due to it indicates that after pollination the color keeps on changing in these flowers. Okay, see there another flower I have brought you. It is a inflorescence. Okay, here you can see the flower very clearly showing. Bi-lipped corolla it is having, green calyx it is having, and it is bearing the anther towards the outer side. Okay, can you see yellowish color? Even you can see it in tulsi. Okay, these are the flowers of tulsi. The whole thing we are going to call it as an inflorescence. Okay, that yellowish color what are emerging out, those are nothing but those are androecium. Slowly when you are going to complete this chapter, wonderfully you people are going to learn lot many things from nature. Okay, which you can implement in your life. Okay, there is another flower which you people are most familiar with. You people can also go through your gardens, or you can plant some plants. Okay, you people can learn lot more things, what all the beautiful thing creature has made us to learn. Okay, when we are going to dissect, we are going to understand everything. For being kind, I'm not going to dissect all the flowers, only one flower I'm going to dissect for you people, and I'm going to show everything. Today we'll be starting with sexual reproduction in flowering plants. Okay, are we not lucky that plants reproduce sexually? Okay, the mites of flowers that we enjoy gazing at, the scents and perfumes that we swoon over, the rich colors that attract us are all there as an aid to sexual reproduction. Okay, what a beautiful scent we are going to get from the flower, and what a beautiful colorful petals we are going to see. Those all are not for sake of us, they are sake in order to attain the sexual mode of reproduction. They are going to attract the pollinators in this fashion. It might be by the scent or nectar they are secreting, or by their beautiful colorful petals. Okay, so by which the pollinators are being attracted, and this aids the pollination, okay, which further enhances the fertilization. So for our selfishness, we have thought that flower beautiful things are only for us, okay. All flowering plants show sexual reproduction. A look at diversity of structures of the inflorescence, flower and floral plants shows an amazing range of adaptation to ensure the formation of the end products of sexual reproduction, the fruits and seeds. At the end of the sexual reproduction, we are going to get fruits and seeds. This means that the sexual mode of reproduction has been taken place by this thing, okay. So in this chapter we'll be understanding the morphology, structures of androecium, gynoecium, okay. They are told here as the sexual reproduction structure we are going to understand in these flowering plants. Human beings have an intimate relationship with the flowers since a large amount of time, okay. So it might be due to the aesthetic values they are having, ornamental, social, religious and cultural values they are having. We are using these flowers as the symbol of conveying important human feelings such as love, affection, happiness, grief and mourning, okay. Much before the actual flower is seen on a plant, the decision that plant is going to flower has taken place. Several hormonal and structural changes are initiated, which leads to differentiation and further development of the floral primordium. Floral primordium means a bud of flower, okay. The plant initially does not actually start with flowering. There is some phase of vegetative growth, after which there is some hormonal changes in the plant. So the plant also bears starts to bear flower, and also there is hormonal and structural changes in the plant, and then the flower primordium starts to appear, okay. Inflorescences are formed which bear the floral buds and then the flowers, okay. First buds are formed. From buds the buds are going to mature into flower. In the flower, the male and female reproductive structures, the androecium and gynoecium, differentiate and develop, okay. Androecium is nothing but the male reproductive organ, which we call them as stamens, and female gynoecium we are going to call it as a female reproductive structure. So friends, now we'll be studying with the male reproductive organ. We are calling them as a stamen, microsporangium and pollen grains. Friends, if you people are convenient with the diagram over the book, you people can refer that, or if you people are convenient over here, you people can understand by seeing this natural image, okay. Now I'm going to take it. Okay, what we are going to see, the yellow colored part, what we are seeing, what we are observing is nothing but the anther, the yellow color one, this one large yellow color one, and this small one what we are observing is a filament, okay. Now we'll come across anther shows the two parts of a typical stamen. The long slender stalk called as filament. This long slender stalk what we are seeing is a filament, and the terminal general bilobed structure is called as anther. This bilobed structure we are going to call it as an anther. The proximal end of the filament is attached to the thalamus. See, we are going to call this end of anther as proximal. You people can understand, proximal, and this end we are going to call it as a distal. So proximal end is attached to the thalamus. Thalamus just now you have seen in the flower, okay. This bulged portion where the pedicel of this flower is being increased, okay. For this pedicel, the calyx, corolla, androecium, gynoecium are attached. In some cases what happens, anthers remains attached to the petals. The proximal end of the filament is attached to the thalamus, or in some cases it might be attached to the petal of the flower. The number and length of the stamens are variable in the flowers of different species. So you can study all these things. See, now the filament is so small in this flower and anther is so large. So this will not be same in all flowers. So you people can see, dissect some flowers around your nearby the home environment, and you can study all these things, different kinds of stamens which are varying structurally. So carefully observe observation of each stamen under a dissecting microscope, making the neat diagram would elucidate the range in the shape and attachment of the anther in different flowers. Okay. Now please listen carefully. A typical angiosperm anther is bilobed, with each lobe having two theca. See, here is one lobe and here is another lobe. Can you see? One lobe and another lobe. They have taken the cross section of this anther. This is one lobe and this is the another lobe. Okay. These two lobes are, now we are going to call such an anther as bilobed, with each lobe having two theca. See, one theca and another theca, one theca and another theca. Each lobe is going to have two thecas. Okay, they are dithecous, bilobed dithecous in condition. Often a longitudinal groove runs the lengthwise separating the theca. See, can you see this longitudinal groove which is going to separate one theca and another theca? The various types of tissues and their organization in the transverse section of anther. You can see a large number of tissues are there. In this small amount of space, there are large number of tissues. One by one we'll be studying in a future class. Okay. Let us understand the various type of tissue and their organization in the transverse section of the anther. The bilobed nature of the anther is very distinct in the transverse section of the anther. The anther is four-sided tetragonal structure. See, 1, 2, 3, 4. Four-sided. So we are going to call that term as a tetragonal structure consisting of 4 microsporangia. See, 1 microsporangia, 2 microsporangia, 3 and 4 microsporangia. 4 microsporangias are there, located at the corners, 2 in each lobe. See, 1, 2. In one lobe we are having 2 microsporangia. In this lobe we are having 2 microsporangia. Okay. So what microsporangia we have seen now? Okay, those are going to develop. See, 1, 2, 3, 4. In cross section, anther appears in this fashion when we are going to take this section. Okay. So we are going to see 1, 2, 3, 4 microsporangia. In this microsporangia, microspores are going to develop, and which microspores in later cases they mature and become pollen grains. Okay, you people are getting? See, microsporangia develop further and become pollen sacs. Okay, they are telling about pollen sacs. See, now understand. This is sporangia, microsporangia. Okay. For this sporangia, we can also call when mature pollen grains are present. In that case, we can call it as a pollen sacs. They extend longitudinally all through the length of anther and are packed with the pollen grains. Okay, throughout this anther, what you people have studied. Okay, now anther was in this fashion, bilobed condition. Within this anther, what happens? The pollen grains are present throughout. When we are going to take section, it appears in this fashion. Okay. The structure of microsporangia in a transverse section. A typical microsporangia appears near a circular in outline. It is generally surrounded by 4 wall layers: the epidermis, endothecium, middle wall layers, and the tapetum. See, now you people are understanding. See now. Now you people understood how we got this sporangia. Okay, very detailed. I will tell you once again. See, this is an anther which is having bilobed structure, and this is an filament. So when we are going to take this section at this region, anthers section at this region, what happens? So we are going to view in this fashion. When with the help of microscope, when we are seeing this section, it appears in this fashion. You people are getting my point. So here 1, 2, 3, 4. 4 microsporangia will be there. So to protect this inner microsporangia, there are 4 wall layers towards the outer side. Means when now I am going to enlarge only this part. Only this part I am going to enlarge. See, this is a microsporangia, friends, and this is the outer wall. of that sporangia. Now you people are getting. Now I have enlarged only this much part I have enlarged. So this outer wall is called now as epidermis, where this epidermis is going to protect the inner microsporangia. Here actually the microspores or pollen grains are going to develop. Okay, after epidermis they said that there will be endothecium. So now I am going to draw. Endothecium will always be bit large, okay, at maturity. When you are going to see the mature anther section, it will be a bit large and it will be having some kind of fibrous thickening. But now it will not be having any kind of fibrous thickening in immature stage. And later on there will be a small, very thin walled middle wall layers. Next, what we are going to call them as middle wall layers. And next to this, we are having very important wall called tapetum, which is in close contact with this this tapetum is in close contact with this developing microspores. Okay, please remember that this tapetum is going to nourish this developing microspores. Now we'll name everything. First thing what I called first wall layer was epidermis. The second was endothecium. And the third was middle wall layer, and the fourth was tapetum. Now you people understanding, because it is having very important role, so I have highlighted a bit. Even these two are having important role. Okay, this is going to degenerate at maturity. We can't see any of the wall layers of tapetum or either middle wall layer. So when they are not present, we are going to call them as ephemeral in nature. Ephemeral. So these are the new terms you will be learning in future. Generally surrounded by wall layers. Okay, fine. Three wall layers perform the function of protection. Which all wall layers? I told you epidermis, endothecium, middle wall layers are helping in protection and help in the dehiscence of anther to release the pollen grain. And when anther is going to mature, the pollen grain has to dehisce from the anther and they have to reach the stigma. Okay, there they are. So the term they are calling is dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. I told you. Now tapetum, what it does, it nourishes the developing pollen grains. The cells of the tapetum possesses dense cytoplasm and generally have more than one nuclei. See, I told you people earlier, see this tapetum, as it is in close in contact with this developing microspores. Throughout all these things you can see the tapetum, all this microsporangia, not only for this one microsporangia. So all these four layers are present for all the microsporangia, not only for one microsporangia. Okay, these tapetums are going to nourish this developing microspores within the microsporangia, and they are not only nourishing. In order to nourish, they should have large amount of cytoplasm. Cytoplasm will be larger amount, and the nuclear content will also be more. It might be diploid, triploid, and tetraploid also. Okay, so that large amount, see, if we are having large amount of money, we will be going for trips and all. In the same fashion, if it is having large amount of money, what it is going to do, it is going to give out the nourishment for the developing pollen grains. Okay. When the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue occupies the center of each microsporangium. It means that, see, these microsporangia are not going to directly give rise to microspores, okay? Before to which, here, now we have studied with all these three wall layers, okay? Now we'll be studying about this microsporangia. So I'm going to take only microsporangia outside. So when it is young, in younger condition, all these microsporangia are compactly arranged in this fashion. Okay. When they are going to mature, what happens? They become pollen grains. When later what happens? These compactly arranged cells, they are going to take the globular shape. Now we are going to call this as sporogenous tissue. Okay, at little amount of maturity, we can see the globular appearance of these cells. Okay, and even these are slightly compactly arranged cells. Okay, we are going to call them as pollen mother cell or microspore mother cell. Okay, and these pollen mother cell, now they are going to undergo meiosis. When they are going to undergo meiosis at this stage, what happens? Only when we are considering with only one cell itself will give rise to the 4 cells. Now you people understand? So now you can think of so many large amount of pollen mother cells are there. So all large amount of pollen mother cells, now they are going to give rise to the microspores after reductional division called meiosis. And these microspores, after attaining a kind of maturity, they are going to develop vacuoles with a dense cytoplasm, and they are going to become into pollen grains. Okay, these microspores, now we are going to call them as pollen grains. As the anther develops, the cells of this sporogenous tissue undergo meiotic division. I told you, see, as anthers are developing, as anther, inner side tissue is appearing in this fashion. How the tissue is keeping on changing. Okay, how the anther is going to develop? Initial stage anther will be this fashion, and slowly when it matures this fashion, and slowly when it is going to mature in this fashion, and when it is going to dehisce, it is going to rupture out. All pollen grains are going to come out. Whenever you are going to touch the hibiscus flower, we are going to get yellow color powdery substances, or no? So those are nothing but pollen grains. Okay, in this fashion the anther is going to develop. When the anther is developing in this fashion, inner side the tissues are being developing in this fashion. Okay, Microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrad. Yeah, now I'll tell you what is this microspore tetrad. Soon after the formation of microspores, these microspores, as I told you that one of the pollen mother cell is going to give rise to the 4 microspores. Okay, these 4 microspores are intact. See, they might be in this fashion 1, 2, 3, 4, or they might be in the L form, or they might be in the J form, or they might be in the T form, or they might be in the linear form, or they might be in the tetrahedral form. 3 are facing here and another one is facing in opposite side. So this kind of structures we are going to call them as microspore tetrads. You all understood? We are going to call this structure as a microspore tetrads. So in this fashion, anther is going to dehisce here. Okay, at the time of dehiscence, these spores are going to come out. So here what happens? As each cell of the sporogenous tissue is capable of giving rise to the microspore tetrad, each one is potential pollen or microspore mother cell. The process of formation of microspores from the pollen mother cell through the meiosis is called as microsporogenesis. Now you all understood what is microsporogenesis, where one pollen mother cell is going to give rise to the 4 microspores. This is called as microsporogenesis. The microspore as are formed are arranged in cluster of 4 cells, the microspore tetrads. Now you are understanding the thing. See, they have given in your book the tetrad in this fashion. Microspore tetrad, how they are arranged. As anther matures and dehydrate means when without it is going to dry out, the microspores dissociate from each other and develop into pollen grain inside the microsporangia. Several thousand microspores are pollen grains are formed at the release with the dehiscence of the anther. When anther is going to dehisce, at that time microspores are going to become into a pollen grain and they are dehisced in the form of pollen grain. Now up to here you people have understood. Okay, in next class we'll be studying with what are the pollen grains, how the pollen grain is going to germinate and how it is going to form generative cell and vegetative cell. Thank you friends.